Thank you so much, Kitty. And thank you everyone for joining us today for this special presentation. We're really thrilled to be able to bring this to the group and really hope that you enjoy it. So would like to, at this point, introduce our guest speaker today. So welcome to PKDOC, Professor Meg Jardine. So Meg is currently the director of NHMRC Clinical Trials Center at the University of Sydney, and her research focuses on reducing the progression and complications of non-communicable diseases, particularly kidney disease, diabetes, and cardiovascular disease through randomized trials and wow, I can't talk today. I am sorry. Wow. I'm just going to skip that part. Through randomized trials, and Meg will share more information about that. So again, we are delighted that Professor Jardine has agreed to share her work with us today, and especially given that platform trials may have particular benefits for PKD, where pathological mechanisms may be different from other more common forms of kidney disease. So ways to efficiently generate specific evidence will be important moving forward in the field. So again, welcome Professor Jardine, and we're really thrilled to have you with us today. And at this point, I'll go ahead and turn it over to you.
All right, thanks so much, Laura, and thanks very much for having me in this meeting, and thank you particularly, Kevin, for helping to facilitate this. Today I will be focusing on the trials, so you're right to focus on that, Laura. I think it's really important that we find ways to get the best evidence so that we know we can find the best treatments and use treatments that work, discard the ones that don't. And we need better ways to do that in some of the rarer conditions like polycystic kidney disease. So I think this topic is really applicable to people with polycystic kidney disease. These are my disclosures front up. I've worked with a range of companies on various trials, none specifically on polycystic kidney disease, although some of the trials have been open to people with polycystic kidney disease as well as other conditions. So what I wanted to talk about today is just to outline what adaptive clinical trials are, to go over some of the points in which these trials may be particularly suitable for people with various kidney disease conditions, particularly polycystic kidney disease, touch on some of the success stories from other areas so we know what we can reach for, and then open it to discussion. I'm aware that there'll be a range of knowledge here. Some people will know about adaptive trials, some people might not. So I hope that there's something in there that's of interest to everyone. But I guess what I'd really like from today is the discussion part at the end. I'd really like at the end feedback on what you think this means for people with polycystic kidney disease. Which aspects do you think are particularly of interest? How you see it all fitting in. So this is a slide that a lot of trialists like to show, and what it demonstrates is the growth in trials over the years. And you can see that as we get into more recent years, as we move to the right, there are many more trials being registered, which is great. But the green line down the bottom is kidney disease, nephrology, and you can see that we've got a bit better, but we're consistently the worst. Now, partly that's not a fair accusation, partly it's because we have a rarer condition, but I think it's also because we haven't been able to organise ourselves as well as some other areas in medicine, and that's where these trials might play a role. I think everyone wants to do more trials and get more trial evidence. The ISN, the International Society of Nephrology, a few years ago had a roadmap session where they tried to work out what the challenges were in kidney disease health and how we could try and meet them. And that meeting decided that we should aim to have at least 30% of people with chronic kidney disease in a trial. Now that's a big reach, but if we don't reach, we won't get it. So out of that, the activities of a subgroup of ISN became really important, and that's the International Society of Nephrology Advancing Clinical Trials Group. And this is a group that firstly I should say is open. Anyone in the nephrology community can join the ISN, and you can register your interest in various activities. And so in the ISN Act, we have about 250 people, I think, who've registered interest. The aim of ISN Act is not to perform trials ourselves. It's more to look at what are the barriers to trials and what are some of the solutions. The idea of having an adaptive trial, a platform trial in nephrology has been heavily discussed in this group. So I might just take a moment to outline what I'm talking about, what the difference is between the traditional trials and the trials that these newer formats that we're talking about. So, oops, sorry, just getting on top of the technology here. A traditional trial will design the trial. Everything's decided up front. It then launches the trial according to pre-specified plans and then analyses the trial at the end. And that design's been really robust. But the problem is if you make any errors or there's anything you don't know well at the beginning in the design phase, the trial really can't, is set, it's set in its way. An adaptive trial tries to take all that uncertainty into account. So ahead of time in the design phase, you work out where the uncertainties are and you build in rules that you can adapt as new knowledge comes through. So then when you conduct the trial, you review these various parameters, the uncertain parts, you adapt the trial according to the rules you'd set out and so forth. And then at the end you analyse. Now, what are those uncertainties? Well, things you might not know at the beginning is exactly how strong an effect you might get. So you might not know how many people you need in the trial. In an adaptive trial, you can change and update the sample size according to rules as you go on. You can abandon treatments that don't look like they're performing well or particular dosages of treatments, and that allows you to concentrate on the ones that are looking promising. You can change how many patients are randomised to different arms of the trial. So again, if an arm is looking promising, you can allocate more patients to that arm, and that means you're going to find out whether it works more quickly. You can also look at particular subgroups. There might be patients with different characteristics, different genetic mutations, for instance, and you can see whether various arms are working better in those patients than in others. And the other thing you can do is you can stop the trial at an early stage. And that might be because the treatment is more effective than you thought. And so you've already found that it works. Or it might be because the treatment is not effective and that it's looking futile. And then you can save all the effort and move on to things that are more promising. Now, another concept I'd like to introduce within this adaptive trial space is different types of adaptive trials, and some of you might have heard of these terms, umbrella trials and basket trials. And these are two extremes, and there's sort of everything in between. In an umbrella trial, you'll take multiple treatments for a single condition. And there are a few examples that are already going. The iSpy 2 trial is one you might have heard of, which is an oncology trial in breast cancer based around California. And in that trial, different treatments are being tested, all for women with locally advanced breast cancer prior to surgery. So quite a specific population with multiple treatments. And we'll touch on that later on today. A basket trial takes the other approach. It'll take an intervention, and in our space, that might be an angiotensin receptor blocker or an SGLT2 inhibitor, and test it against multiple diseases. So in our situation, in kidney disease, we might test an SGLT2 inhibitor against in people with diabetes, in people with non-diabetic glomerulonephritis, people in polycystic kidney disease, for instance. So that way you're looking at lots of different subtypes and you can see, is the treatment working overall or does it work better in some groups than others? And all of this is moving us towards a more precision medicine approach. And there's everything in between. And there are many trials that in effect have both a basket and an umbrella approach. So now I'd like to focus this broad concept in on our challenges in kidney disease and how we find new treatments. And we're going to touch through some of these aspects. So I think the first point to make is these trials are ongoing. You establish a platform, you build up all your infrastructure, your recruitment infrastructure, your safety boards, your adjudication boards, and those bodies keep on going. And that saves a lot of a lot of communal energy because you're not re-establishing all the time. It helps sites because they know what to expect, and it certainly helps patients because the structure is there. And one example is the Stampede trial. Now, it was set up as a platform for prostate cancer in the UK, and it was set up in 2005, and 15 years later, it is still going. So that means for patients in that area with prostate cancer, they don't have to wait for a trial to be set up. There's one there. And that one's now recruited over 10,000 patients. And I'd like to show you how this sort of works. So what you're seeing in this graph is the sort of history of the Stampede trial. And down the bottom in the green, that's slowly rising. If I move my mouse, can you see it or not?
Yes.
No, okay. Yes, okay. So this trial, this line here is the first trial. And you can see that that looks a fairly typical recruitment curve. What you're looking at is the number of sites in the study that had recruited at least one patient. And on that trajectory, it would have taken 9 years to recruit 120 sites, that opened 120 sites that had recruited at least one patient. So that's a typical slow story. And then you can see the next trials. There's the yellow one, which was one stopped early, and then in the other colours you see the later trials. And you can see how quickly they got to the point with sites recruiting at least one patient. So most of them, by 6 months, they had 100 sites with at least one patient in that particular trial. So what that's telling you is the power of infrastructure. Those sites had everything basically set up and they just needed to open the arm with the new trial. Now we're looking again at Stampede, and now we're looking at patient recruitment. And each of these dots represents a patient recruited. So up here, A, that's the first trial they ran, starting at the end of 2005. And you can see a slow start with a few patients being recruited, and then gradually more and more patients being recruited till we get to 2018. And all the trials, that the 6 interventions they started at the beginning of Stampede, all had that slow start and then built up. But now we come to the more recent trials. Here's one starting around 2011, 2012, and you can see how dense those dots are, and that's telling you how quickly the patients are being recruited into those trials. Now, partly it's because the whole Stampede network was growing, and partly it's that infrastructure at the site level. So that's the efficiency that I think we should aim for. So a second characteristic of these adaptive clinical trials is they mostly have a single primary endpoint. So this is a sort of schematic of what a trial might look like. You start your trial, you have a control arm, you have a few different investigational agents. After a while, one of them might prove to be not effective and is stopped, other ones come on, and the whole infrastructure keeps going. But what pins it together is they're all answering the same question. And in our area, it might be progression of kidney function. So can we do this in nephrology? And I'd argue we can. We tend to over and over use the same endpoints. We're repeatedly, frequently looking at the amount of protein or albumin in the urine and at changes in kidney function or changes in EGFR. And I've listed here some of the trials that we have in nephrology. This is as of 2019. And you can see all the different conditions we've got: C3 glomerulopathy, chronic kidney disease that isn't otherwise characterised, diabetic nephropathy, and so forth and so forth. And then in the columns, you can see how many of them are looking for these variants on the same endpoints of albuminuria, proteinuria, and change in kidney function. So in effect, we are using the same endpoint. So that reduces the barriers to a platform trial where we move all these individual trials into one common structure. And even within those broad areas of albuminuria, we tend to focus, or proteinuria, we tend to focus in on the same endpoints. So what I'm showing you here is the FSGS trials and the endpoints they used. Now, most of them look for remission, and they define remission. That's the disease, in effect, turning off. They define remission in terms of the protein in the urine, and they use very similar endpoints. So to my mind, we don't have a big barrier there. We are very close to having a single common endpoint and can probably reach agreement on that. The next characteristic of these trials is shared infrastructure, and I've spoken about that a bit, so I won't spend too much more time on it. But just to emphasise again that establishing all these components of a trial takes time and energy and cost, and if you set it up once, you're saving on efficiencies there. The next characteristic is the multiple agents, and we've talked a little bit about the advantages there. In the Stampede trial, this is the agents that they had tested. So they started with 6 agents, standard of care against various different potential interventions, and more have come through. So you can see how many, we're up to 12 already that have been tested when this slide was produced. Another example is the iSpy trial. That's the one of breast cancer based in the US, in the south, and I'm going to get my geography, west of the US in California, thank you. And what this is showing you over time is the agents that they've tested. So they started down here, and I'm not going to try and repeat those names, and have very successfully completed a number of trials. What the colour coding tells you is the outcome. The ones in purple have what they describe as graduated. And in iSpy, what graduation means is that you've proven the agent works and it leaves this phase 2 trial and moves on to larger testing. In the green, agents that have been completed but maybe weren't successful. But that again is important to know. You don't waste any more time on that, you move on. And then there are a couple of trials that have been halted. What this shows is the overlap, and I think that's really important for the enduring infrastructure aspect. Before any one agent had stopped its trial, the next one was in there, and generally they've had a few going at once. And so this means quicker time to finding treatments, and it also means options for patients who are keen on participating in trials. Now, one of the things that makes this model efficient is the shared control arm. And what that means is that instead of testing 5 different agents in 5 trials with 5 control arms, if they're all tested in the same trial, you have 1 control arm against the 5 interventional agents. That means that you can recruit faster because you don't need as many patients to recruit. And what it also means is that for any patient coming into the trial, they're more likely to be randomised to an active agent than to control. And generally that's been, most patient groups report that that's an appealing characteristic. Now, I think why that's particularly important to nephrology is because we have a condition that's often rare, or at least patients with the condition are spread out, which makes the conventional model challenging. What these numbers show is some of the incidence of some of our conditions in terms of 100,000 people per year. So, for instance, amongst adults, membranoproliferative glomerulonephritis occurs at 0.2 people, that's 1 in 5 people, per 100,000 per year. And the other conditions are similar. They're all about at most 1 per 100,000 per year. So that means we need something different from the standard approach to be able to be successful in completing trials. One particular challenge is in the area of FSGS, and I think this has really come to attention because there's been so much industry interest in FSGS, and we have more and more agents coming through. But if we don't manage to set up structures that allow us to test them, industry will stop being interested and we'll move on to other indications. All up, for the completed and the ongoing trials that are already registered in FSGS, we would need to find a little over 2,000 people to successfully complete just the ones that are registered, let alone the ones that are coming down the pipeline. For the active trials, there's a recruitment target of 1,400 people. So that means of all that we've ever tried to do, we're trying to do 65% of it now. So we haven't succeeded in rapid recruitment in FSGS in the past. We now have an even bigger challenge. So we really need to be organised on so many fronts to deliver these trials efficiently. All right, now I'd like to change tack a little bit and talk about the statistics. I know some of you will be more interested in statistics than others, but I hope what I can convey to you is some of the efficiencies in this design. So what many of these trials will do will start with fixed randomization. So for instance, in concept, you might have a trial with a control arm and 3 active agents that are being tested. So when we launch that trial, you might start with 1 to 1 to 1 to 1, or 1 in 4 people randomized to control and 1 in 4 people randomized to each of the active agents. And then you let the trial run. You get to a planned interim analysis, and that's this red line here. And at that point, you look at how patients are responding. And that might be on a surrogate marker, or it might be your primary endpoint. So in the breast cancer area, often it's progression of, it's the size of the lesion on imaging, how big the lesion is and whether the cancer has shrunk. For us, it might be the amount of protein in the urine, or it might be early changes in the EGFR. So you look at those changes, or I guess in polycystic kidney disease, another good marker is the size, the kidney volume, and that's been used in a number of polycystic trials. So at this interim, you look at how people are going. And what you might find is that, that people assigned to agent 2 look like they're responding better than others. So from that point, you haven't got proof, so you let the trial keep going. But from that point, you put more patients onto agent 2. So you adjust your randomization algorithm. You still don't know for any particular person what they're going to be randomized, but overall you're going to get more people randomized to agent 2. And the trial unfolds. And then at the next interim, you do the same, and it might happen that again, agent 2 is looking more promising, and so more people are randomized to it. So you can see that over time, what's happening is we're getting more and more patients randomized to agent 2, fewer and fewer to agent 1, which wasn't looking so promising, and to agent 3. You always need patients randomized to the control, or you're not able to definitively prove. And the effect of having more agents onto agent 2 means that you've got more and more power to assess whether agent 2 is working. And so you more quickly arrive at that point of definitive proof. Now, what your level of definitive proof is has been decided at the beginning of the trial. What the adaptive randomization is doing here is getting you there more quickly. So you more quickly find that the promising agents work or don't, and it takes longer to find out if the less promising agents work. But that doesn't really matter because they're less promising. So finding out they don't work is not such a priority. Finding the ones that do work is the most important priority. So I'm going to take you through a real example here of how this would work. And this is for, again, a trial from a cancer condition. And this was a pretty rare condition. So they were only looking for a smallish trial of 75 patients. And what they wanted to do is look at the standard of care, which is shown here in the orange, against 2 experimental arms. And these were all combination treatments. So they start the trial and they randomise evenly. 1 in 3 gets randomised to the standard, and 1 in 3 gets randomised to each of the intervention arms. And then they go through this process of assessing response. And what they see over time is that the blue arm, the second experimental arm, isn't going so well. And so fewer and fewer people are randomised to that arm. And we get to the point here, after 24 patients, where it's met the predefined definition of futility, where they said at the start, if it's looking this poor, we're going to abandon it. And that was reached after 24 patients. So they stopped randomising patients to the blue arm and then randomise everyone else to the control and the first experimental arm. And in this design, they decided once they'd dropped an arm, they would just completely randomise according to how well the patients were going. And so you can see most patients get assigned to the control combination, and the trial stopped after 34 patients. The eventual, the analyses, the definitive analyses, showed overall patients randomised to the standard combination succeeded, met their definition of success 56% of the time, and this was in a rare cancer. Patients randomised to the first experimental arm were only successful 27% of the time, and patients randomised to the second one, weren't ever successful. What you can also see is that more than half of the patients, 18 of the 34 patients, were randomised to standard, which turned out to be the best arm. And what you can also see is that the trial finished, it had reached all the predefined definitions of proof and futility after 34 patients, whereas the standard approach would have given them 75 patients. So in less than half the time, this trial answered some important questions. And then one last point I'd like to make about the statistics is the power we can use when we're looking across rare conditions. And that's so often the case for us in nephrology. And in this schema, for instance, you had lots of different patient subpopulations randomised in a trial. And it could be anything. For us, it'd be different types of nephrology conditions. In cancer, it might be different types of mutations. And this will be, this is a situation you might use when you've got really rare conditions. And what you then do is empirically look at the end of the trial and see who's responded to what. So you might say this group in blue are all the subconditions that responded to the treatment, and the ones in orange are the ones that haven't. In any one of these groups, you wouldn't have had the power to answer the question, or you might have had a trial that ran for years. But by looking for patterns amongst responders and non-responders, we start to get an indication of where the treatment is likely to be most effective. It's going to have other kick-on benefits as well, because once you know that there's something in common between these conditions, between the blues, you start to have better insights into what the pathology is in some of the rarer ones. And that's going to be really useful for nephrology, because there's so many conditions we have where we don't understand what the pathology is. So getting some insights and knowing what behaves similarly might help us answer some of those questions. So I'm going to just conclude with a couple of the big success stories. Recovery, who I think, I hope everyone's heard of. Recovery is the trial running mostly in the UK. And it was an adaptive platform clinical trial that was set up really early at the beginning of the COVID pandemic. There were other things that made recovery a success. The health infrastructure was there to support trials, and the Department of Health in the UK just decided, they were committed and that this was going to run in multiple hospitals. But part of the efficiency was the platform. They were able to test multiple treatments in the platform. When one worked or didn't work, they were able to spread that knowledge, but keep on putting more and more treatments through the platform. So by August, which was less than 6 months after they started in March, they'd recruited over 12,000 participants, and they're still going. And they're active in 176 sites. They've been able to show things that didn't work. The lopinavir-ritonavir combination didn't work, so that arm was closed because it didn't work. The low-dose dexamethasone arm, that's been closed because it did work. And so they were able to get those results out quickly. Those results have been taken up all around the world. They were, in the meantime, they carried on. So they showed that hydroxychloroquine didn't work, and some other trials have shown that. And on they go. But what's really exciting is the timeline. Starting in March, hydroxychloroquine answered in June, dexamethasone answered in June, and lopinavir-ritonavir again by the end of June. And these are some of the results, but I won't dwell on that anymore. So in summary, as I see it, the potential benefits for running these master protocol or adaptive trials in nephrology are firstly, the infrastructure means that more patients will get more access to different research agents through trials. Secondly, it's going to be much more efficient. We're going to generate more evidence and generate it more quickly. We'll be able to look at rare conditions that we might not be able to test in isolation. And by including patients with rare conditions with patients with other conditions in a trial, we'll be able to get insights into how those conditions might respond and indeed what their pathology is. Because of the shared infrastructure, there'll be lower costs for trials, and that again means there's more, hopefully more funds available for trials. But I think most importantly, the efficiencies mean that the trial questions are answered more quickly, and that means beneficial treatments are coming into the market more quickly, and agents that are not beneficial or perhaps might even be harmful can be discarded more quickly. And the third thing that these trials allow is they allow you to incorporate external learnings along the way. And that's the adaptive nature of it. So if there are other trials going on at the same time, that are showing evidence, you can incorporate that into the trial design rather than waiting for everyone to finish and then find that the answer was obvious. So a big key to making this all work is working together, and that's moving away from the competitive structures that we often have in medicine. We often have different groups competing against each other, setting up trials that compete against each other, to turn all that energy into a joint exercise and make it easier for people to collaborate than to compete. And we'll do that if we've got an efficient structure that allows everyone to work together. So the goals, coming back to the ISN Act, what we felt the goals for an enterprise like this would be to firstly build that infrastructure, to build a network of people with chronic kidney disease that are willing to participate in trials. Not everyone is, of course, but to build that network. And so that then when trials are available, they can be launched. We already know who might be interested. You don't have to sort of launch a trial and then put in a whole lot of energy into trying to find people who might be interested. And in particular, to embed in this structure, these are adaptive clinical trials, which would mean we would hopefully harness all of these efficiencies. In kidney disease in particular, I think we need to think global, and I guess as an Australian in a relatively small population, maybe that's really clear to us. If we don't work together, it's going to take a long time. With a rare condition or a low incidence condition, we really should be working together. So that was all I wanted to share formally in slides. What I'm really interested to know, though, is to get feedback on how all this sounds, and in particular to get feedback on what are the things that are appealing about these types of design. Is there anything that's not appealing? Is there any area where we should be looking for new solutions? So they were the three things I'd really like to hear back. But I'll turn over to you, Laura, is it? And you can organise it.
Yes. Yeah, that's great. Thank you so much for this talk. It was really, I think, enlightening for this group to hear a little bit more about it. So we do have a couple questions coming through the chat, so I'll go ahead and start there, and certainly to the group, feel welcome and in fact encouraged to send some more questions through as we would, definitely like to take advantage of this discussion time. So one of the first questions that came through was a question that was surrounding something that you talked about early on in your presentation, which was when you shared the graphic on the umbrella and basket trials. So the question was, are the umbrella and basket trials typically conducted in already marketed drugs?
The answer is sometimes yes and sometimes no. So sometimes you'll get a drug that's really shown to be very effective in a large population. For us, we might think of diabetes. Many of our trials have been conducted in diabetic kidney disease, but then we don't actually know if the agent is going to work in people with other types of kidney disease. So that would be a really good setting for an adaptive trial to test in all those people who weren't in the original big trial. And an example, for instance, the SGLT2 inhibitors. We really know they work effectively in people with diabetic kidney disease. There are some groups where we don't know if they're going to work. We know we have a little bit of evidence from the DAPA-CKD trial, but we don't know, for instance, if they work in people with polycystic kidney disease who are excluded from the big trials, and many other groups. So that would be a great example where we could do an adaptive trial. That was
Great,
a long
thank
answer.
you.
I'll try and get my answers quicker.
Oh, no, no problem at all. That's a really great information. So we have another question here from Alan Yu. Thank you for the outstanding talk, first of all. So the question is, how do you work with the multiple companies that are each developing their own agent? Seems like they would need to relinquish control of the study design, conduct and analysis to the trial network center. How do you handle that?
Yeah, look, that's a great question. And that's, I think it's that competition collaboration axis that we really need to get to. So when there's nothing established, companies will want to run their own trials as they've been used to. Where we do have effective adaptive trials established, because they are quicker and more efficient and already have a setup and they've got they're able to communicate and access patients to go into the trial and sites, companies will go to what's most efficient. And we can see that playing out in the oncology area. There are a number of adaptive trials in the oncology space. I talked about iSpy too, but it's one of many. Because they are working well, companies are prepared to relinquish that control you're talking about to go into what's a successful enterprise. It needs to be coherent. So you need to have be using an endpoint that everyone agrees on. There are a few other things, but in general, in nephrology and other areas, our trials in many ways look very similar. We use similar endpoints. We have similar trials of similar lengths of time. We use similar types of standard care. There's a lot that's similar. The points of difference are relatively small.
Great, thank you. Our next question is from John Michael Sauer. He's the Executive Director of PKDOC here at Critical Path Institute. So his question is, do we know the appetite of health authorities, US FDA, EMA, PMDA, TGA, for these umbrella or basket trials in the nephrology space? Have they been approached for feedback on these types of trials?
Yeah. That's a great question as well. Generally, the regulatory authorities are familiar with this design, again, from the oncology space, which has led the way. In nephrology, I think they are receptive to hear from us, but yeah, and there are some smaller adaptive trials that have been used. Often companies will run small phase 2 trials with different doses, for instance, using adaptive models. The appetite, I think, will be strongest for the early discovery, the phase 2 trials, as we move to, and we see that in oncology as well. For the larger trials, probably there will always be a space for the standard trial that we're familiar with. The regulatory authorities will certainly embrace this design, but like every trial, you always would approach the regulatory authorities with your design and look for any trouble points ahead of time before launching. And that's a process we go through with the traditional model as well.
Great, thank you. So our next question comes from Elise Hoover. When making a case for funding, what strategies do you use to estimate cost, since the protocol is expected to change so frequently?
Yes, aspects of the protocol change, but one of the important things is that all of this is predefined. It's not a free range, do what you like. You work out the things you don't know and what you're going to adapt on. And that's things like early response and number of events, for instance. But what it does mean is that you don't know, typically you don't know how many patients you're going to need to recruit, what the eventual sample size is. And mostly it's less. Sometimes it'll be the same if you've got very few agents, for instance, and if they're not effective. But the more effective the agents are and the more you have in them, generally the fewer number of patients you need. So yes, you don't know your budget, but your budget might be lower than you expected. So most people can wear that risk of coming in under budget.
Always helpful. Okay, so here's the next question from Kevin Fowler. Can you share the learnings from oncology pharma companies and investigators on how barriers to adaptive trials were overcome?
I think so. The biggest barrier is the startup, because it's new. and people will tend to go with what's already in the marketplace and what's working. So establishing a master protocol trial or an adaptive trial is the biggest challenge. I think the way the oncology networks have done it is they've essentially, as I see it, they've leveraged off existing networks that were people who were and sites who were already working together, and within that launched an adaptive trial. You tend to see the effective ones they've got something shared. It might be a region shared or an existing infrastructure or a data system. So I think for us, that's something to really think about. We need to think about shared structures that help us to work together. One of the things that's successful is in effect that like-mindedness. And I think this is a place where patient organisations might play a particular role, because patient organisations tend to be not so concerned about different institutions and different companies even. The focus is really on what matters, which is completing trials and getting more agents to market. So I think the patient voice is really important in this space to say, get yourselves organised and work together.
Right. And here's another question that kind of follows up a little bit on the infrastructure piece. Assuming that it's cost and time intensive to establish, the infrastructure for some of these larger master trials, who covers the cost for that? Is that something that is ISN ACT is doing, or does that fall on industry and patient organizations to come together to collaborate on that cost to set up the initial infrastructure?
Yeah, that's a really good point. That's maybe that's the biggest challenge. It's getting it started and getting the seed funding to get it started. Once it is started, and we see this for trials like Stampede and for iSpy, because they're successful, the profits that would normally just go back into the organisations fuel the ongoing platform. So it's a self-sustaining model as long as you've got new treatments to try. For us in nephrology, we need to get that start up. And ISN Act itself is not a company. It's not and ISN isn't a company. What we are looking for there is seed funding, and we're looking to industry for that. But also looking at possible academic sources to get the seed funding to establish the network, and then it should sustain itself until we've run out of treatments.
Okay, another question. How are multiple dosing schedules normally handled?
They can be relatively straightforward, actually, because you might have just one company involved. So you will, in those examples where I had multiple arms, each of those arms might be a different dosing schedule. So you might not know at the start, exactly which dose is going to be most effective. So you might start, with say 5 milligrams, 10 milligrams, 50 milligrams of a dosing schedule of the one agent. And then as you run along, according to your preset rules, you would look at how patients are responding, and things that maybe were looking not effective or harmful, you would prune off. You would stop randomizing patients to those arms. So it would mean that the ineffective or the harmful dosing schedules get abandoned early instead of having to wait to the end of your trial.
Great. Okay, so the next question that's come through, many of the examples you've shown for adaptive trials were diseases in which the required follow-up time, the clinical outcome was relatively short. For example, advanced prostate cancer or breast cancer, or time to remission for FSGS or IgAN. An adaptive trial structure is also being used to test COVID treatments in the US. What are the challenges in applying this trial design to relatively slowly progressing chronic diseases such as PKD?
That is a brilliant question. Yeah, that is really insightful. So congratulations, whoever thought of that. That's really true. They do work quicker. In fact, most trials work quicker when you've got short time to information. A conventional trial is quickest in that setting. For these ones where we want to adapt according to response, it will work best when you're able to read that response early. And you're exactly right. In the breast cancer, it was the size of they were adapting on the size of the tumor before surgery. So that was, you know, after 4 weeks or after 12 weeks, you can measure the size of a tumor. And similarly, COVID and so forth, all the examples you gave. That does create a challenge for slowly progressive conditions. And that's something that nephrology is grappling with more generally. So there are two solutions that are the most commonly spoken about that I've heard. One is to use the response to albuminuria. That's the amount of albumin in the urine. Where people have high levels of albumin in the urine, those levels drop relatively quickly with an effective agent. And we've seen that with the angiotensin receptor blockers and the ACE inhibitors a couple of decades ago. And more recently, we've seen it with the SGLT2 inhibitors and some of the other conditions being tested, atrasentan, for instance. So if you have albuminuria in the first place, you can get a readout on that within 3 months. So that's one solution, and you can then use the response to albuminuria to adapt on. That's not going to apply to everyone because not every condition involves albuminuria. So the other strong contender in the space is really the change in eGFR. So instead of waiting until someone's had a doubling of serum creatinine or needs to start dialysis, you look at how their eGFR is changing. That's their creatinine, essentially, how that's changing on blood tests. And change in eGFR is being put forward as a trial endpoint even for conventional designs. Now, it still is a relatively long readout. You know, you still need, say, 2 to 3 years at least before you've got a good response on eGFR. So eGFR is probably not going to be a good way to adapt on, but albuminuria potentially is.
Thank you. Okay, so the next question, if a treatment arm in an umbrella-style adaptive trial proves effective, how do you maintain the control arm on a now expiring standard of care, i.e. how to maintain clinical equipoise in the control arm in light of new information constantly emerging from the adaptive trial?
Yeah, look, really good question. So it's all about deciding at the beginning and committing yourself to what your burden of proof is. And we do that with conventional trials. You know, we decide that we have to get a p-value of less than 0.05. So you decide with this, when is it proven, and when would you be in a situation where ethically you'd want everyone to have access to that treatment? And so then as the trial unfolds, when you get to that point, that's when you declare. That's when you've met your predefined standard of proof. And at that point, it depends really what your access is. You know, one model is that at that point, everyone moves on to that treatment and you keep testing others. So you've got a new standard of care, and that you continue testing other novel agents, but as background treatment, everyone now gets the new standard of care. That was one of the early lessons. There was one of the very early adaptive trials was set up and they didn't have that model, and a new standard of care came in, and they didn't, you know, then of course they had to abandon the trial. So now generally the trials will have that if something's proven and available, that becomes the new standard of care. That it isn't always available. Sometimes that agent then has to go off to the regulatory bodies, and so there might be, you know, a delay. But if it is available, for instance, if you're testing something that's already in the marketplace, that then becomes your new standard of care.
Thank you. Okay, so the next question from, and I'm sorry if I mispronounced this, Terry Christoph from Galapagos, Belgium. Thank you for the very interesting talk. As a sponsor, who will choose to stop or to increase the integration of patients for testing the sponsor's drug in such adaptive trials?
So this is, again, it comes to the collaborative approach and deciding these things up front. And so generally what the adaptive trials do is once you're in, once your agent is running in the trial, every agent is treated in the same way. So if the patients on that arm are responding better to the surrogate or the interim, then more patients are randomized to that. And that decision is agnostic of the particular agent. You treat all the agents the same. Another decision point, though, maybe you're interested in is how do you decide if you get into the adaptive trial? Say you're terribly successful, which is we're not. We talked earlier about the challenges of setting up. But once you have set up, if you're working well, maybe many sponsors would like to use that infrastructure and put agents to the trial. And these are principles that have been outlined in some of the iSpy publications. Generally, what these trials have, they'll have some sort of structure for selecting agents in the trial, and they'll have some sort of rules around or criteria around that. So they may be about an unmet need, and they'll also maybe be about the capacity in the trial. If depending on the size of your trial and the number of patients coming in, maybe you don't want to have more than 4 agents in at any one time. So there'll be rules like that, criterion-based rules, and like many things, they're best if they're decided according to transparent criteria up front.
Great. Okay, it looks like we have just enough time for the last question that's come through. For a basket type trial, are there multiple control groups given that different diseases are being evaluated?
Generally there's one combined control group, and the different diseases will be coming through. We'll have some patients assigned to control and some assigned to elsewhere.
Okay, great. So those are all of the
maybe
oh,
so
sorry,
from the point of view of someone with one of those rare conditions, if you're going into a trial with 4 agents in control, you've still got roughly 4 on 5 chance of getting assigned to an active agent, just like anyone else coming in.
Great, thank you. Those are all of the questions that have come through so far. So again, we really thank you for spending your time today with us. We really appreciated your talk. Are there any last minute questions from the group that you would like to pose before we wrap up today?
Can I ask one?
Absolutely.
Yeah, look, I've heard lots of questions and the same questions that I get from clinicians and everyone else. Is there anything about this from the patient perspective that is particularly good or particularly bad? Perhaps particularly bad. Are there any problems from the patient perspective?
Hey, Meg, this is Kevin. I don't see any negatives. I think the challenges in kidney disease is that the expectations have been really low. And so this is just like a quantum leap for this community. I think your comment that you're making about just like sharing data, right? Those are areas that I don't think the patient organizations are engaged in. And I think if they were, I think you might start to see some change.
I think that's a good point, Kevin.
The other takeaway too, I think just the path of least resistance that you identified is just thinking about where you have networks where there is collaboration and then focus upon where people are working together. And that's my other takeaway. So thank you very much.
Good point. Look, if there are any thoughts that occur, maybe if they could send them through to you, Laura, because it'd be really great to get that feedback on the things that are particularly appealing and whether there are any barriers or problems.
Specifically from the patient perspective? Perfect.
Yeah.
Yeah, absolutely. If there are any further questions that the group has after digesting some of this information, please don't hesitate to send them in to either myself or Wendy Vanaska, who wasn't able to join us today, but usually works with this group. And we'll make sure to pass those along to you, Meg. And then also conversely, as Meg just mentioned, if you have any thoughts on, potential barriers or thoughts from the patient perspective, or any information that you would like to give feedback on, please send that in to us and we can coordinate getting that back to you. So
that'd be fantastic.
Great. Well, thank you again so much for your time today, and thank you everyone for joining us. We were thrilled to be able to offer this special presentation, which is a little different timing from our usual group. So we thank everyone who is able to join us today. And again, we're happy to facilitate some follow-up discussion after today. So thank you everyone for your time.